Hello everyone, welcome back. Today we'll discuss the new gene editing technique CRISPR-Cas9. CRISPR, formerly known as clustered regularly interspaced short palindromic repeats, represents an intriguing family of DNA sequences found in the genomes of prokaryotic organisms, including bacteria and archaea. In the last century, it became clear that these sequences play a crucial role in the bacterial immune system and offer a glimpse into the complex defense and counter-defense structures that occur at the microscopic level. Each sequence in the CRISPR assembly contains a palindromic repeat, which is a mirrored pattern of nucleotides that carries a person's unique fingerprint. These particles form the basis of the bacterial defense mechanism in immunological encounters. The main element of CRISPR is Cas proteins, which serve as the protectors of the cell. Guided by the information encoded in the CRISPR sequence, these proteins, with unparalleled precision, cut invading viral DNA with remarkable efficiency. Recognizing the potential to expand this CRISPR-Cas system beyond the bacterial battlefield, researchers have repurposed this elegant mechanism for a transformative purpose: gene editing. This formal approach implies a conscious shift from observing natural processes to actively harnessing their power for the advancement of human knowledge and potential medical breakthroughs. For this tool to reach its full potential, ethical considerations, potential off-target effects, and societal impacts must be carefully examined and addressed. By delving deeper into the mechanisms and applications of CRISPR, this formal research aims to provide a comprehensive understanding of this groundbreaking technology and pave the way for responsible and ethical use of this technology in shaping the future of gene editing. Now let's take a deeper look at the new gene editing technique CRISPR-Cas9. The interest in understanding and modifying the human genetic code has increased considerably with the emergence of CRISPR-Cas9. The gene editing technique inspired by the immune system of bacteria is a breakthrough that offers unprecedented potential for transformation in medicine, agriculture, and our understanding of biology. As we said at the beginning of the video, the acronym CRISPR refers to a family of repetitive DNA sequences found in the genomes of prokaryotic organisms. These sequences, which initially did not attract much attention in the bacterial genome, later turned out to be the basis of the immune system. Bacteria use CRISPR-Cas9 adaptively, storing fragments of past viral infections and using them to recognize and neutralize future attacks. Recognizing the importance of this natural memory, scientists have repurposed it for gene editing, transforming it from a defense tool into a tool for rewriting the code of life. Let's start by talking about the meaning of Cas9. Maestro is an enzyme that acts as molecular scissors, able to cleave DNA with impressive precision at sites designed for RNA. Guide RNA, or simply gRNA, is a special RNA molecule designed to match the target DNA sequence, directing Cas9 to the desired location. When the gRNA directs Cas9 to the target DNA, the enzyme causes a double-strand break. The cell's natural repair mechanisms are eager to restore harmony and thus offer two potential solutions. Now let's move on to those two solutions. Non-homologous joining, or NHE for short, is rapid, but the repair process can result in random insertions or deletions at the break site, potentially disrupting gene function. This is often used to study gene function. Homology-directed repair, or HDR, is a more precise repair process that uses a donor DNA template to guide the introduction of desired genetic changes. This method has impressive potential to correct disease-causing mutations and rewrite the malady of life. CRISPR-Cas9's unique advantages, sensitivity, and ease of use and affordability have placed it at the forefront of gene editing technologies. Its potential applications cover a variety of fields with a lot of possibilities. Now let's move on to the areas where CRISPR-Cas9 can be used. Let's start with gene therapy, which can correct genetic mutations responsible for diseases such as cystic fibrosis, sickle cell anemia, and muscular dystrophy, offering hope where there was once only despair. Cancer immunotherapy is another important topic. By engineering immune cells to recognize and attack cancer cells more effectively, we create a victorious finale for this formidable foe. The last possibility we'll talk about is personalized medicine. Tailoring treatments to individual patients based on their unique genetic makeup ensures that each person receives the most appropriate treatment for their health. Now, let's move on to the subject of agriculture. We can ensure a productive harvest for everyone by developing products that are more resistant to pests, diseases, and environmental stresses. Increasing the nutritional content and shelf life of food products creates a nutritional symphony for the world. We can also create designer crops with special properties for different environments, ensuring food security in a variety of climates and biomass. There are many other fields that CRISPR-Cas9 can. Understanding biological processes will enable us to study the effects of CRISPR-Cas9 can have. The development of biofuels and other sustainable technologies creates a harmonious future for our planet. Conservation efforts continue to resonate through the preservation of endangered species and their genetic diversity, providing the richness of life's melody. While CRISPR-Cas9 offers a dazzling area of possibilities, its power requires responsible use and careful consideration of the ethical and social consequences that resonate like discordant notes. Germline editing will create heritable changes and raise complex ethical and social questions about the nature of humanity. The potential to create designer babies with desirable characteristics raises concerns about eugenics and social equality and jeopardizes the symphony of diversity. Ensuring that everyone has equal access to this technology, regardless of their socioeconomic status, is of great importance in preventing the continuation of social inequalities. So will CRISPR-Cas9 be the new technique for editing genes? The gene editing environment is preparing for a very important transformation under the leadership of CRISPR-Cas9. However, it is safe to say that this revolutionary technique has many advantages over the existing methods and has become arguably the best in the field of gene editing. In conclusion, despite CRISPR-Cas9's remarkable sensitivity and versatile applications in the fields ranging from medicine to agriculture, its status in gene editing remains dependent on the responsible development and application. The future of gene editing will likely involve a collaborative approach. In which CRISPR-Cas9 harmoniously complements other techniques according to their strengths and limitations. And that is it for today. Thanks for watching. As always, it was Hair Cell Times.